Good morning. My name is Elizabeth Rogers, and I am a graduate student and scientist in training at the University of Missouri. To put it very briefly, my research involves trees and pollution. And two of the main investigators besides myself that are involved in this research are Dr. Chung Ho Lin of the Center for Agroforestry and Dr. Ronald Zalesny Jr. of the USDA Forest Service. Before I begin and get into my research, I would like you to visualize something. So in your mind's eye, picture a landfill. I want you to picture all of the contaminants that exist within and around the landfill, whether they're in the soil, in the groundwater, in the runoff. And around that landfill, I want you to picture trees being planted intentionally, trees that grow very fast and have extensive rooting capabilities. What you are visualizing right now is represented in this photo right here. This is a photograph of one of our field sites. We have extensive rooting trees with rapid growth, hybrid poplars, planted next to a landfill. We have 16 of these sites established in our overarching project that I am a part of. And I would like to talk about my objectives as well in this project. My main goal as a researcher in this is to help expand what we know about hybrid poplars that are planted at landfill sites like this. The first phase of my objectives is to collect information on the ecophysiology and on the ecosystem services of these poplars at landfills. One of the main parameters that I'm interested in is water use. How much contaminated water are these poplars taking up as they're acting as biological pumps? When they grow, they take up so much water. I want to know how much water they're taking up, whether that's daily, weekly, monthly. In addition, I am interested in what particular compounds are in the contaminated water and which ones are being taken up more. Are there trends among clones? Once I have this information collected, I'm going to synthesize it and integrate it into a format that is relevant for different groups of people, whether that be researchers or site managers or the public, even including K-12 students. One of my passions is education. In 2019, I have had some preliminary accomplishments regarding these objectives. For one, I have begun my sap flow literature search. So I'm looking at the sap flow of poplars, whether that's at landfills or just in the forest. I have also begun to establish a leachate pollutant prioritization scheme, and I've collected some samples of leachate and groundwater from these sites, and I'm learning in my laboratory training how I will be analyzing these samples. And finally, I've established my experimental design for field instrumentation and data collection, which I will be starting this summer. And just to leave you with an image, we began with an image, we'll end with an image. Every city that's on this map, aside from Rhinelander with the star, has one or more of these hybrid poplar landfill sites. The ones that I will be focusing my research on are those that are circled in green. And I've given you an image of each of them, along with a date that the image was taken, just so you can visualize and really see how fast these trees actually grow. Thank you very much.
Hello, my name is Ben Schuessler. I'm a first-year master's student here in plant sciences, and my project is looking at post-harvest handling practices of the black walnuts. My academic advisor is Andrew Thomas. The project is in conjunction with the Missouri State University with Chin Feng Huang, and Dr. Ron Revord is also on the project. The objectives of this research is to explore the effects of drying duration on seed viability. Viability is assessed by a tetrazolium assay as well as a germination study. Another part of the project is to assess the accuracy of a float test to sort out viable seeds from non-viable seeds. This is also tested by a tetrazolium assay. So preliminary results of the drying study show that between drying seeds for 2 days to 12 days, seed viability decreases the longer you have your seeds in drying. And it seems, as the graphs show, there is a weak correlation between mass and its likelihood to succeed. Preliminary test results for the float study show that those that sink immediately are likely to be viable, whereas seeing the ones that initially float were floated for another 24 hours to assess whether or not they are truly viable or not. As you can see here, the ones that floated essentially twice are not viable usually, and those that sank after being floated for 24 hours had a variable success rate. Preliminary conclusions of this project show that viability decreases as drying duration increases, and there's a weak correlation between viability and mass depending on the drying duration. And the float study conclusions show that it is reliable in sorting viable seeds from inviable seeds, and less reliable when assessing those that floated and then sunk. Thank you.
Hello everyone, my name is Vân. I'm currently a PhD student emphasis in forestry. My advisor, Dr. Lin. And my research is focused on identification and characterization of the health promoting benefits of the black walnut, and that can promote the novel use of the black walnuts and its byproducts like dietary supplement or functional foods, skin care products or personal care products. And this project will provide opportunity to turn abundant low-value material and renewable material into the high-value added products in the Missouri. And we characterize health benefits of 22 black walnuts selected for the nut production and and by the Center of Agroforestry. We found that the black walnut have the strong anti-inflammatory, antibacterial, and antioxidant activities. The biological function of the black walnut very variable among the cultivar tested. Several health-promoting bioactive compounds responsible for the antibacterial, anti-inflammatory, or antioxidant were identified and characterized. Many of them are novel bioactive compounds, and for example, we reported the first time of the antibacterial activity of the Gagenian A, responsible for the antibacterial activities, and also this compound is very abundant in the black walnut. Our approaches is combine the analytical chemistry and molecular biology, metabolomics analysis for the phytochemical study, and the approaches we use for the black walnut study can apply for another land material, including the elderberry, sweetgrass, Oasis orange, black walnuts, or the spent coffee round. And I think for the identification of the phytochemical can allow us to turn the low-value material into the high-value products. And with that, last year we published a couple of the paper, and we had one book chapter and several the manuscript in preparation. And we have the poster in the hallways. If you're interested, can stop by and look at it. Thank you.
Good morning, everyone. My name is Priyanka Sharma, and I'm a PhD candidate in natural resources. I'm working with Dr. Sogata Burden. And my PhD research was mainly focused on the effect of biochar on soil properties and biomass yield in marginal soils under tree and grass-based systems. It's a long topic. That's what PhD is. So it's a long process. Anyways, so biochar is basically a material that is created by burning biomass at high temperatures in oxygen-limited conditions. So for my study, I basically created a marginal soil. So there are many studies out there that have focused on the effect of biochar on agriculture soil. But my purpose was to create marginal soils so as to look at the effect of biochar on marginal soils. So for my soil, I created a soil mix that had 80% sand in it. And also there was a component from the soil that I added. So the objective was to investigate the effect of biochar and nitrogen application on soil chemical properties. So for soil chemical properties, I mainly looked at important soil nutrients like phosphorus, potassium, carbon, nitrogen, and other soil properties like pH, cation exchange capacity, and electrical conductivity. For microbial parameters, I looked into soil microbial abundance and microbial diversity. And then I evaluated nitrogen use efficiency by plants, and then biomass production, above ground and below ground biomass. So the two plant species that were grown from my experiment were eastern cottonwoods, that's Populus deltoides, and switchgrass, that's Panicum virgatum. So the purpose of choosing these two plants for the research study was, first, these two crops are leading biofuel crops. Second, they thrive very well in marginal soils. So that were the two reasons I chose these plants. Now, my experiment was a potted experiment conducted in a shade house for a short duration, for one growing season. Unfortunately, I did not see any significant effect of biochar on soil properties, which were like chemical and microbial. But total carbon showed a significant improvement on addition of biochar. So it showed linear increase. As I increased the rate of biochar, the total carbon in the soil increased linearly. So biochar did not have any significant effect on soil chemical and microbial properties except total carbon, as I've already said. So, and also, there was no significant effect on biomass yield and nitrogen use efficiency. On comparing two plant species, the soil under switchgrass showed better soil properties than the soil under poplars, which leads us to the conclusion that switchgrass production in marginal soils may be more sustainable than growing poplar in marginal soils. Also, because my experiment was a potted experiment, the potted conditions may not be a true reflective of how soil amendments would behave under field conditions. And also because it was a short-term project, like short duration project, and biochar is mainly known for its positive impacts for a long duration. So maybe the duration was not enough for the positive impact of biochar to become apparent. But since it had a significant improvement in the soil carbon, and soil carbon has very important role in ameliorating soil conditions and other soil properties. So maybe we can expect to see more positive impact in long-term experiments. Thank you.